Hello, hello, hello. Hello, everyone, to this Burlington House Lunchtime Scientist talk. This is actually the sixth talk in our astrobiology series. And today I am quite sad because my usual co-host, Lucinda Offer from the Royal Astronomical Society, is not with me. So I'm hosting this alone. I'll try and fill her giant metaphorical shoes, and Lucinda will be back next week. My name is Joe, and I work at the Linnean Society, which is a natural history organization. It aims to inform, involve, and inspire people about nature and its significance through our special collections, our programs, and also our scientific publications. This series of talks was co-produced, as I said, by the Linnean Society, where I work, and the Royal Astronomical Society, where my usual co-host Lucinda works. The Astronomical Society is an organization that encourages the study of astronomy, solar system science, geophysics, and other closely related branches of science. Our two organizations can be found in Burlington House, a great big building in the center of London, alongside some other cultural organizations, including the Royal Academy of Arts, the Royal Society of Chemistry, the Geological Society, and the Society of Antiquaries. Our two organizations have decided to co-produce this series for young people with an interest in astrobiology because it's kind of an overlap of our two interests. I'm excited to pass over to our speaker in just a moment, but before I do, I just want to say that this talk is being streamed on Zoom webinar, so you might be in a Zoom screen right now. And it's also streaming live onto YouTube, and it might be on some other social media sites if that's being shared. If you are watching this live and not sometime in the future, you can type questions or comments into the Q&A boxes either on Zoom or on YouTube, and we'll try and incorporate them into our conversation at the end of this talk. Our speaker today is Dr. Alex Price, who is an environmental microbiologist. Alex studies whether Mars could be habitable or could have been habitable in the past or the future. However, I don't think Alex is planning to go to Mars to study this. Alex is here today to talk about analog research and what that means for astrobiology. So without further ado, thank you for joining us, Alex, and I'll hand over to you now.
Brilliant, thank you very much. I'll just share my presentation. Is that working properly?
Perfect.
Brilliant, okay. Thank you so much, Joe, for that introduction. That was excellent. So my name is Alex Price. I am a microbiologist at the Open University down in Milton Keynes. I'm an early career researcher. That means that I finished my PhD quite recently. I'm a bit earlier in my career than some of your other speakers who've talked in previous weeks, such as my colleague, Karen Olsen-Francis and Silvio Sinibaldi, who talked about planetary protection as well. So what I'm going to do is I'm not going to go so broad as those other speakers. I might just focus down a bit onto what it's actually like to be an analog researcher and a microbiologist working in astrobiology day to day. So I will just, yes, that's working. Okay, great. So I'm part of Astrobiology OU, which is the astrobiology research group at the Open University. You might have heard of the Open University through either our adverts or just through friends and family who might have already studied with us. We're mostly a distance learning organization, which is what you might have heard of before, but we do have an actual research campus as well, where our PhD students, postdoctoral researchers and academics all work together to advance science and research in different areas of academia. So our group itself is actually now the biggest dedicated astrobiology group in Europe. We've got over 50 researchers, students and support staff who work together in Milton Keynes. So it's great to be part of such a sort of broad group of people who all have different interests and different research interests as well. And that really helps in a field of astrobiology. So what exactly do we do as a group? We cover a whole load of stuff, including space governance, which is the laws and rules that govern how we explore space. Things like commercialization, turning space technology into businesses, International development, what can space technology do to promote international development? Education, how can we use astrobiology to actually teach science, because it's so multidisciplinary, there's so many people from different areas working together, that it's a great tool to actually teach different areas of science and technology in broader society as well. But mostly I'm going to be focusing today on this bit, the science, because that's what I do in my day-to-day. But even the science of astrobiology is really broad. And that's kind of the whole idea, is that astrobiology isn't a defined field of science in the same way that something like microbiology. So all microbiologists study microbes, all chemists study chemicals, all astronomers study the stars and sort of cosmological objects as well. So it's not that, it's centered around a set of common questions, and we use various different strands of science to actually approach those questions. So NASA defines it as the study of origins, evolution, distribution, and future of life in the universe, which is an incredibly broad topic. But it can pretty much be boiled down to sort of three overarching questions, which is, how do we get here? That's talking about things like the origin of life and the history of life on our planet and possibly other planets in our solar system. Are we alone? That's this level of life detection, going out to places like Mars to actually look for traces of life in the universe, and also on a higher level, looking for sort of the idea of SETI, sort of communicating with possible extraterrestrial intelligences, although that's not what I really cover. And where are we headed? What's the kind of future direction that we're going into? So that's got stuff to do with planetary protection when we sort of spread out into the solar system. How are we going to protect those environments from us? And how are we going to protect ourselves from what might be there in those environments as well? So on the first of those questions, I just thought I might be polite and introduce myself a little bit. Joe did a good job at the start. But my personal origin of life was up in Newcastle upon Tyne. And then I've gone through a sort of variety of different science subjects to get to where I am. So I started off doing my bachelor's in biomedicine, so not really anything to do with astrobiology at all. But then I sort of moved into microbiology and then eventually managed to get a PhD project within this research group that I work in currently. So that's one thing to say really early on, is from a career point of view, if you're thinking about what you might do at university, you're really interested in astrobiology, there's not really sort of like an undergraduate course or a master's course in astrobiology that'll get you a degree in that. What you do is you train yourself in scientific disciplines, and then when you become a researcher, you work together with people from other disciplines to actually conduct astrobiology research. So yeah, that's a little bit about where I've been, but you might think that the way that sort of scientists get to where they are is very much a straight line, as it kind of looks on my slide here, that I just did a bachelor's, master's, PhD, all the way through to astrobiology. As I said, I wasn't, at your age, I wasn't set on being an astrobiology researcher. I happened to do something I enjoyed at university, which was the biomedical science. And then the techniques from that, actually, when I became interested in astrobiology, really helped me get into microbiology and then to apply those methods to astrobiology. So that's a key thing to say early on, that from a career point of view, you don't have to go in an absolute straight line. Loads of people who I work with in astrobiology have taken really circuitous routes through their careers and ended up exactly where they want to be eventually. Okay, so enough about that. What are we actually talking about today? So I'm talking to you about analog research. And maybe the best thing to do to start there is to define what we're actually talking about in astrobiology when we talk about analogues. So analogues defined as a personal thing that's comparable to another. And that gives you a hint as to what we're talking about with analog environments in astrobiology research. So we're thinking about areas and environments on Earth that have some crossover with a particular extraterrestrial location that we might want to investigate for signs of life or life itself. So these are places on Earth that have particular characteristics where they cross over in a certain way. So it could be something like pH, So quite often we go and look at really, really acidic environments, like you can see in the top left there. That's Dalol in Ethiopia, where the waters there are really, really, really acidic. But around the margins of these really, really acidic pools, we find microbial life. And that's a common theme that runs throughout our sort of understanding of life on Earth, especially over the last few decades. We've come to understand that pretty much everywhere on Earth where it's possible for life to exist, life does exist. So in another way, things like aridity, a lot of analog research goes on in places like the Atacama, the driest places on Earth, which are very similar to what Mars is today, very dry, and sometimes sort of cold and very hostile surface environments. As I mentioned, temperature, we look at the hottest and coldest places on Earth and what are the actual limits that life can survive in those places. So if you look to the top right of these images, that's actually a black smoker at the bottom of the ocean. So these are hydrothermal systems there where volcanism is actually heating up water in the seabed and driving this hot water up through these structures into the ocean. And around these, the water can be superheated to well over 100 degrees Celsius because of the pressure. But around the margins of these, once we get below about 120 degrees Celsius, we find that there are active and living cells around here, microbial cells, but also once it gets a bit cooler, a bit further away from these, you get large things like tube worms, and certain types of crab can actually live in these really hostile deep sea environments. So we know that life is really robust in the deep ocean. Another thing is salinity or saltiness, basically. So we find microbes that are living in sort of salt deserts and salt deposits across the world, especially archaea, which are called halophiles, which means that they really love salt. And they're actually evolutionarily adapted to live in these really, really salty environments without their cells being compromised, like ours would be. It could be the underlying geology of a place. So in the bottom left there, you can see some acid mine drainage, which is really rich in dissolved iron. And that red color that you see in the water is actually iron oxide, so kind of rust forming as bacteria actually metabolize this iron and actually use this iron for their energy supply, essentially. So microbes can be really adaptable, and different species of microbes can do incredible things in really harsh environments. And final thing is oxygen availability. So pretty much, a lot of life on Earth depends on oxygen. We do, clearly, and pretty much all complex life does as well. But there's a lot of microbes that can actually survive and grow and thrive, really, in anoxic environments. So we know that the early Earth was quite anoxic, and we think the same of Mars. It doesn't have a lot of available oxygen in the atmosphere. So we might think about types of life in sort of anoxic environments, in sediments on Earth, that might have some crossover with conditions on Mars as well, or conditions on Mars in the past. So there's a really broad range of analog environments that we go out to in various places on Earth and conduct astrobiology research. So those are all what are called natural analogs. These are all places we go to that have this crossover. But there's also another side of analog research, which is a lot more of what I've done throughout my PhD and up until now in my current position, which is talking about simulated environments. So this is exactly what you would expect. This is taking what we know about a place like early Mars or current Mars, maybe the icy moons. And what we do is we try and recreate those conditions in a laboratory setting. And then we test the ability of Earth microbes and different kinds of life that we have on Earth to actually tolerate and survive under those conditions. So I'm going to take you through a bit of my PhD research and talk to you about simulated environments. And then later on, I'm going to talk to you about a natural analog that I've been looking at as part of a collaboration with one of my colleagues, Savi Philippidou, who's another postdoctoral researcher at the university. So we're going to talk about a natural analog in Botswana later on. But first, I'm going to take you through some simulated environment stuff. So this is Mars, obviously. And when astrobiologists are thinking about life on Mars, there's really two big overarching questions that we're trying to ask here, which is, could life have existed there? This is the habitability question. Is it possible that life could have existed given the conditions on early Mars? And then the second question is the life detection question. Did life exist there? Can we go there and find traces of either ancient life or much more recent life in situ at Mars? So could it have supported life? Did it support life? So the best place to start looking at this, as I've already mentioned with the analogues, is what we know of life on Earth, because it's the only place that we know for sure in the universe has life. So it makes sense to use life on Earth as our kind of benchmark to start investigating this. And it's really useful to look at the history of Earth and Mars together to see what we're kind of interested in here. So you can see on the left-hand side that when you compare the ancient Mars, so we're going from over 4 billion years ago on the left-hand side of the image here to the present day on the far right. So on the far left of here, you've got a crossover of a few 100 million years in early Mars's history, where you have a lot of liquid water, you have a dense atmosphere, you have quite a warm environment as well, and you have a lot of volcanism, you have a lot of volcanic energy, a lot of chemicals being dissolved into these environments and creating loads of energy supplies for potential life if it got started on Mars. So that's one of the reasons we talk a lot about early Mars, is because that's where it's got closest crossover to the history of life on Earth. So within this early Mars environment, we're looking between about 4.2 and 3.7 billion years ago. So there's a few 100 million years called the Noachian period, where, like I say, you've got this warm, wet environment. And you can see here that there was still volcanic activity or widespread volcanic activity. That puts a lot of greenhouse gases into the atmosphere, which, you know, are seen in a negative light in the current circumstance of climate change caused by us on Earth today. But in the early Earth history and early Mars history, those gases are really important for maintaining a thick, warm atmosphere that allows liquid water and liquid oceans to form on the surface of these two planets. So you have this crossover of these kind of anaerobic, anoxic, but quite warm and wet early environments. So we know that life got started on Earth in that environment. The question is, did it get started on Mars? So we need to think about, as microbiologists, thinking about this, what directions would life take on early Mars? If it did get started, what would it actually use? What would it look like? What should we be looking for when we send missions to go and detect life on Mars? So coming back to that, we're really looking at this area at the start, this pink sort of circled area. But what you'll notice if you look along the bottom is that the only life on Earth at this time that you can see along the bottom is prokaryotes. So prokaryotes are microbes. They're bacteria and archaea, the two domains of life that are purely microbial. So those are cells that don't have a cell nucleus. Eukaryotes that have a cell nucleus, like us and like all complex life and like fungi and things like that, didn't come in till much, much later on, billions of years later, in fact. And that's after Mars had already become quite a hostile, dry, cold environment. So we think that if life really got going on early Mars, it probably wouldn't have had a chance to become little green men or anything really complex. We're thinking mostly about microbial life, like small single cell organisms. Another key point is that evolution follows the environment that life finds itself in. So it's going to use the energy sources that are available in that environment. It's going to use the oxidants that are available in that environment. So that just means, so we use oxygen as an oxidant, for example, but there are plenty of other ones that microbes can use. So they can use types of iron as oxidants. They can use sulfates as oxidants. They can use nitrates as oxidants as well. So there's a lot of variety and adaptability in microbial life that we don't have in complex organisms like ourselves. And the third thing to think about is carbon sources. So like, where are they getting their food from, essentially? So what is the carbon on Mars? What was the carbon on Mars in the past? That's a key question as well. So we get ours from food, obviously, but a lot of organisms, plants, and a lot of microbes get their carbon directly from CO2 in the atmosphere as well. So thinking about what are microbes going to use as an energy source on Mars? We know that Mars is the red planet, and the reason for that is that it's covered in iron oxides, rust, basically. But if you dig just a little bit below the surface, you'll find that actually a lot of the iron there isn't rusty at all. It's in another state. It's in a different oxidation state called iron 2, which is what microbes use to get electrons and to sort of feed their energy metabolism and really get growing. So it's a really good source of energy for microbes, and we have it abundantly on Mars. In fact, we have twice as much on Mars per kilo as we do on Earth. So these are the kind of microbes that you might imagine with iron oxidizing or iron utilizing microbes on Earth living in this sort of acid mine drainage. You might have seen photos like this somewhere. So that's one place that iron oxidizing microbes can exist, but all of these ones use oxygen. And like we said before, there's not a lot of oxygen on Mars. So that led me to look at a very particular weird group of organisms for my PhD, which were called nitrate-dependent iron oxidizers. So as the name suggests, don't get bogged down in the really technical terms, but it basically means that instead of breathing oxygen or respiring oxygen, they're respiring nitrate instead. Nitrate is something that you find in like fertilizer very commonly on Earth. We have lots of it locked up in the ground beneath our feet. So organisms that can actually respire nitrate and then use iron as well would be really useful in a Mars setting, because we now know that there's nitrates at the surface of Mars. And in fact, there's abundant nitrates compared to a very small amount of atmospheric oxygen. So looking at these iron oxidizers that don't need oxygen could be really, really interesting. And also some of them can use CO2. So that kind of works out the carbon source problem as well. So anyway, we take these organisms and we basically work out how this would actually work in an early Mars setting. So this is a really complex diagram, but I'll kind of walk you through it. So if you look to the top left, you've got the early Martian atmosphere. There's a lot of carbon dioxide, there's a lot of nitrogen. And then through various processes, this stuff gets processed through lightning, through irradiation, and through impacts as well from meteors to form these nitrates that deposit at the surface of Mars. And then you've got this volcanic activity in the early Mars setting. So that's causing a lot of iron to get dissolved into the water on early Mars. So you've got the iron in the water, you've got the nitrates forming that are available there, you've got CO2 in the atmosphere, and we now know that there's loads of complex organics on the Martian surface as well. So you have food for these microorganisms as well. And that's basically the hypothesis of how these organisms could work on early Mars. So how do we actually go about doing that? What's the kind of like, how does this work in practice in the lab, these simulations? So I use what are called microcosms that are basically little bottles of Mars. So part of that, what I've been doing is looking at whether these microbes can grow on the kind of rocks that we have in the Martian bedrock. So these iron-rich kind of volcanically produced rocks. So things like olivine. So basically you can take a powder of this and basically test the ability of organisms to grow without oxygen on this sort of Mars type mineral as a simulation of how life on Mars might actually act. So what I found was basically that these organisms over a long period of time, we're talking like 100-day experiment here. So these are really slow growing. But what we found was 3 of these organisms that I tested could actually do this and could actually grow on those. But not just that, they also do something really interesting, which is when iron becomes really abundant in the environment, as you'd expect on early Mars, some of these cells start to form these iron oxide rust particles, and these rust particles get stuck to the surface of the microorganisms as they grow. It doesn't happen to all of them, but it happens to some of them, that they become entirely encased in these little rusty jackets of iron oxide. This is something called biomineralization, which is the production of minerals by living things. And what we think is that these encrustations, if they happen to cells on early Mars, you're forming little iron oxide jackets that are going to live, they're going to stay a lot longer in the rock record than the microorganisms themselves. So even if the microbes die off over a long, long period of time, we might still have these encrusted iron oxide traces to look for in the rock record, which would be really cool as well. So another way of simulating Mars is to look at what kind of actual fluids and sort of lake environments and stream environments and groundwaters might have been around on Mars in its early history. So what I do there is I create basically a simulation of what we think those waters would be like. And the way we do that is to take information from actual missions and actual samples of Mars. So in the top left, that's a Martian meteorite. And by looking at this Martian meteorite, geologists can work out exactly what kind of fluids and waters may have passed through that rock when it was forming millions and billions of years ago. And we do the same on Mars as well. So in the top right corner, that's a place called Paso Robles. So that's a really, that was investigated by one of the Mars exploration rovers back in the early 2000s. It took a big scoop and you can see that really bright white yellow material. So that's sulfur. So we know that there's really sulfur-rich environments on Mars. And by looking at the chemistry from that site, we can work out what kind of water was flowing through there before Mars became this dry, barren place as well. And we do the same thing with hematite-rich area that has these little weird blueberry-type structures made out of hematite. And also what we call a contemporary Mars brine. So this is based on the bottom right image. That's Rocknest at Gale Crater. It's where the Curiosity rover landed in 2012. And it's where we've sort of first definitively found what is a habitable past environment on Mars. So we found this lake bed that was actually a sort of a very kind of hospitable place for microbial life to have existed in Mars's early history. So we have a brine based on that as well. So what I did was to take the information that we were getting from that, and then some colleagues of mine turned that geological information into sort of a recipe almost for these brines, and then I turned that into a microbial media to actually grow these in little bottles. So there's a few steps to this, but it's basically linking actual places on Mars and actual investigated environments to sort of direct experiments and research, which is really cool. So this is what they looked like in person. There's a variety of colors showing you the different chemistry and different components that were in these kind of brines. So that was kind of exciting too. And what we found were that some of these organisms were able to grow quite well in these brines, albeit at a really slow, really low level. So they're not producing a lot of cells, but they're kind of they are producing more cells. They are growing and dividing, which is really exciting. And we also found that adding organics to this really boosted it as well. So the discovery of organics a couple of years ago on Mars, or the discovery of complex and confirmation of complex organics on Mars, is really exciting from a microbiology point of view. So yeah, the takeaways from that were that in my experiments, these organisms seem to grow under different relevant Mars conditions. So I'm just going to breeze through this next bit because it's not quite so astrobiology relevant, but it was a big part of my PhD, and I want to make a point here. So what I did for part of my PhD was to look at how these particular organisms, how their metabolism actually works on a sort of, on a deeper level, on a kind of genetic level. What's the sort of genetic component for this? Is it deliberate? Is it accidental that they're actually performing these kind of conversions of like one type of ion to another, or is it just a sort of side consequence of their metabolism? So I looked through sort of genomic data. I sequenced some genomes of these organisms and compared them together to try and find sort of common genes that might explain how this is actually happening. Didn't come to much. But this second experiment that I did was to work out whether or not it was directly related to enzymes that these are actually producing and working with, or whether it was actually just a sort of, they're using a metabolism and the sort of ion oxidation is just something that happens on the side because of the byproducts. My point about this is that I used a knockout study, which isn't something that is widely used in astrobiology, but it's something that's very widely used and developed in medical sciences and in biomedical sciences specifically. So there's techniques that I'm employing during my PhD, like recently, that actually I learned a lot about these from doing quite a different subject for my bachelor's. So if you're interested in going into a career in astrobiology, even if you start off in something very different, the methods that you learn and the things that you sort of pick up along the way can become really useful later on as well. So yeah, the upshot of those experiments was essentially that there didn't seem to be a common pathway of how this actually worked in these organisms. So we think there's different mechanisms for how this actually happens, even on Earth. So there's big knowledge gaps that we still need to fill in our understanding of microorganisms on Earth, as well as thinking about how microbes might work on other planets as well. So there's loads of knowledge gaps to be filled in the future by hopefully some of the people watching today. So the reason that all of this stuff is really important is that it guides what we're actually looking for with life detection missions. So we've got things like the Perseverance rover, the NASA rover that's just landed at Jezero Crater, which is this ancient delta deposit on Mars. It's had all of this water flowing through it in the past, and it's now dry, but we think it was a habitable environment at one point. So we're now sending rovers and instruments that can actually try and detect some of these traces of life. But the research that we do on Earth, the simulation studies and analog work, really informs what we're meant to look for. So should we be looking for traces of particular ion oxidizing microbes? There are plenty of other ones. Methanogens that live in the deep subsurface on Earth and various other places as well. Organisms that grow using sulfur species as well. So but they all form different traces. So doing all of this work tells us what we should be looking for when we actually send the rovers there. So yeah, I don't know if you've been following it, but the Perseverance rover is my current favorite, I think, just purely on the basis that they took a helicopter. So we've had the first powered flight on Mars was part of this mission as well. So if you haven't seen that, it's really cool. Go and look it up. But the Perseverance rover is just getting underway with a lot of its studies, and there'll be some really exciting research coming out of that mission in the next few years. It's not just the Americans that are in on the act. We've also got ESA, the European Space Agency that the UK is part of and remains part of, that have built the Rosalind Franklin rover. So that one's going to be setting off next year and going to a place called Oxyoplanum on Mars that we also think could have been a habitable environment in the past. And that is a life detection mission. It is going to be looking for traces of life in these environments. So there's a lot of really exciting missions coming up around Mars as well to keep an eye on. And taking a load of cool instruments to detect water under the surface, to look at really complex organics that might be left behind by microbes, to look at the geology and the geochemistry of these sites on Mars, so we can learn about the geological history, what's actually happened to that environment, how did it become the way it is, what did it look like in the past, all of that kind of stuff. So it's going to be a really exciting mission. So now I'm going to talk for a little bit about a Mars analog site, a natural analog, that I've been collaborating with some other people on, which is the Makgadikgadi pans in Botswana. So this is what's called a paleo-mega lake, which basically means it used to be a gigantic lake that covered a lot of the southern central African continent. And over time, this has dried up and become less and less hospitable. And therefore, we see it as an analog for that transition zone in Mars's history. So going from this warm, wet early period to this cold and dry current period, there's a transition zone called the Hesperian, where everything's getting drier and more concentrated. All of the lakes and sort of watery environments are sort of drying up and becoming really sort of hard to get to and much less hospitable. So we see this kind of big drying up lake system in Botswana as a kind of analog for that. So it used to be this huge lake, and over the last few million years, it's been, or last few tens of thousands of years, sorry, it's been drying up, leaving behind these gigantic salt pans that are some of the biggest on Earth. So we went there. So I'm going to talk about kind of how it actually works, sort of going to these field sites and investigating them. So we had an original perfect plan that we wanted to go there and sample all of these places that are starred on the map across this transect and look at how microbial life might respond to being at the really dry fringes or at the really sort of the wetter kind of center of the pan where the water pools during the rainy season, and how that transition could be used as a sort of idea of how drying out processes affect the kind of the different microbes you find in these environments. But we soon abandoned that because we realized that these pans are tens of kilometers across, and in the rainy season, when they become wet in the middle, they're very difficult to get to. So we had to switch to a plan B, which was to sample around the edge, still getting this kind of transition from wet to dry through these salty environments, but on a much smaller scale. So yeah, we sort of set out there, and as you can see, it's not the easiest environment to work in. This was day one, and I think that kind of set us up for how difficult it was going to be to work in this environment, because it looks like a sort of solid salt lake that you can just drive across. But actually, during the rainy season, the top of this is very soft. It's kind of a soft mud for about half a meter down. So it means that if you slow down when you're sort of trying to get to your sample site in the cars can get really stuck, and you might get stuck in the middle of nowhere. So that was a learning experience on the sort of first day. And then when we got there, this is sort of what we were doing out there, was going to these locations and taking cores. So we use this core equipment that you can see on the right-hand side. And this basically takes 30 centimeter sections going down through from the surface all the way down. So we managed to take cores that were between sort of 1 meter to 2 meter in depth. And that really gives you a historical record of thousands of years of the pan having rain, drying out, having rain, drying out, and each cycle deposits new material on top of that. So you get these kind of salt pans building up over thousands and thousands of years. So by looking at a core through this time, and by sampling for microbes and looking for microbial fossils as well through this time, you can look at how the changing conditions have actually affected how microbes might behave and what microbes you might find in these cool environments. Working in 38-degree weather in the middle of the day, you try and find shade wherever you can. So that mostly involved hiding under vehicles for a couple of hours in the middle of the day until it got cool enough to do some work again. Yeah, one thing that we don't really think about or don't really talk about too much is that life on Earth can complicate things when you're doing analog work. So if you're looking at an environment that's like Mars, on Mars, you're not going to have any further kind of contamination or inputs or anything like that. If you're working at a Mars analog field site on Earth, the likelihood there'll be things like birds. We had flamingos and cows around the periphery of some of these smaller pans. So there's going to be a lot of contamination coming from current living organisms that you wouldn't expect to have ever occurred on Mars. So that complicates things together, and you have to kind of target what you're doing to account for any sort of more recent inputs or any kind of contamination that might come from other organisms that kind of fall outside of what you're thinking about. Some things were quite a lot like Mars. So I don't know if you've seen the start of The Martian, but we went to this place here called Kufu Islands, and there was actually a dust storm rolled in, and we had about 4 minutes to try and get from where I took this photo. We had a few minutes to try and get all of our equipment into the back of the car before the dust storm took out all of our visibility, and we would have been kind of stranded in the middle of nowhere. So it's an exciting place to be. It's also doing analog research is like, you have to kind of think on your feet because conditions can change quite quickly in a lot of these places, and not always in ways you'd expect as well. So we were also looking at gases. We're really interested in what gases different microbes give off, and how that kind of how the gases that they give off relate to the type of microbes that are there. That's really important for Mars because we're detecting things like methane and other carbon carrying gases that could be produced by microorganisms on Mars. So to understand what organisms give off in these similar environments on Earth, what they're producing into the atmosphere in terms of gases is really important as well. Yeah, so it's not the easiest place to work, and yeah, you can always help out your teammates even if you're just giving them a bit of shade while they're trying to get some of the equipment that's stuck in the ground back out. And eventually we end up at this Ntwetwe pan, which, as you can see, just looks like a whole other world entirely. But there we found these biofilms, which are these really sort of curled up, crusty sort of mats of microorganisms that sort of group together and form these biofilms to protect themselves from the harshness of the environment. And that's the kind of thing that we might have expected if life got going on Mars. When Mars became very hostile, we might expect loads and loads of these kind of biofilms to start forming in these harsh environments. And finally, just, it's a wonderful place. So that's a great reason to go and do analog research as well. So that's me talking about that. Just a few reasons, if you haven't been persuaded already, if I've put you off getting into astrobiology, reasons to get into it. It's really interesting. You'll never get bored working in astrobiology. So it's a really cool area to kind of aim for and to try and get into once you sort of get through university, if you want to get into research, because there's just so many ideas from different areas coming in. You meet people from all over the world. Just in general, working in scientific research is brilliant for that. You meet so many people from different backgrounds with different perspectives, and you get to share your ideas. You get to basically nerd out about space science all the time, which might not seem like the coolest thing to do with everyone, that you know now. But like once you kind of get to my age, you're just kind of like, I just want to talk about the stuff I'm interested in. I love space science. I love microbiology, and I want to talk about those things with other people who are really interested in it as well. You get to go to cool places. I've already mentioned them. There's also really interesting analog field sites in places like Iceland at the top, and you also get to go on things like research visits to other labs and get to exchange knowledge with those people as well. You get to add to the human knowledge of the universe, even in a very small way. So just seeing some of the stuff, some of your ideas kind of published and out there and peer reviewed is really gratifying. It really gives you a sense of kind of job satisfaction. So that's a real plus of working in research as well. And finally, you get to just shout about it. You get to go to outreach events and talk to the public about it. You get to get people excited about it. You get to produce cool stuff like this. One of my friends produced for our group a Lego video that you can go and find on YouTube that explains all of the research that we do. So you get to feature in all of these really cool different things through going and talking to people about your research outside of academia as well. And just to finish on a little bit of shameless advertisement, so Makgadikgadi, or the Astrobiology Graduates of Europe, it's an early career network that I help run with people from across Europe and beyond. So what we do is we organize specific astrobiology events, so conferences that people can present their work at and come along to and see what's being researched across Europe. And anyone can come along to these, even people down at your level. We've had people who are high school students come along just to watch the talks and to really see what current research is going on with early career researchers across Europe. So if you're interested in that, and if you are interested down the line in getting into astrobiology maybe, then go and check out our website. It's got loads of resources and information and places you can look for future projects once you're sort of getting to that point as well. So if you're interested in that, we'd love to have you along to those kind of things as well. So with that, I'll thank you for listening through all of that. It was a wide variety of stuff, and I hope you kind of didn't tune out too much. But I'll pass over to Joe for some questions and kind of open it up to the floor. So thank you for listening.
It's really it's great to hear you talk about your research and I think it's really interesting to me just to think about the challenges that we have trying to understand what's going on in Mars, and how you're particularly approaching that challenge using your analogous environments here on Earth, and also simulated studies as well. It also just seems like a lot of fun. I can just imagine you trying to take pictures on these salt flats without pretending the flamingos aren't there.
Yeah, it's a lot of fun. I mean, as with any career path, it's a lot of hard work, and there's a lot of there's the sort of mundanity of the day-to-day, which is a lot of kind of reading papers and writing that. But even that, compared to a lot of jobs, just reading other people's research, finding out what their ideas are, and kind of writing your own ideas, getting that down in print and sort of seeing that published, as well as the experimental stuff and going out into the field. It's just, it's a really cool environment to be in.
I had a question on YouTube, which is kind of similar to what you were talking about there, which is actually wanting to know more about you and about your research and other research studies in this field. How do people access those?
Yeah. So I would say that there's loads of really good online resources that you can get to if you're kind of at a end of school kind of phase, that I think most of the audience will be. Then there's places like the French Astrobiology Society have actually published a whole series of lectures, as well as these RAS ones and Linnean Society ones, that you can hear a sort of whole range of very distinguished people from across astrobiology talk about their own research. There's just loads out there if you look at if you look for online astrobiology lectures. But in terms of materials and stuff like that, maybe that's probably the best place to start as a jumping off point, because I think even when I was a bachelor's student, that was kind of my first experience of having to read full academic papers, and it's not always the most accessible thing. It's something that you kind of develop your skills at understanding what people are actually talking about in those. But yeah, I mean, the kind of a lot of journals now publish kind of pop science versions of their articles, or kind of lay summaries. So you'll find a lot of the bigger journals now, you can find a summary that's written in like plain English of what this research actually means, and those can be really useful as well.
I actually have so many questions, and I don't quite know how I'm going to tackle these, but I'll just dive in and maybe try and pull some together. One is about conditions on other planets, and whether they might be similar enough to early Earth to lead to this generation of life, or could a different setup lead to a different type of life?
Yeah. So from the point of view of my research, it's very much thinking about how sort of early life on Earth would have developed, and using that as the benchmark. So we look at sort of Earth and Mars together because we do know that they crossed over a lot in terms of the early environments, like I mentioned before, about it being kind of like lacking in oxygen, but quite warm and wet, and having very active sort of water cycles and things like that, and having quite similar geology and rocks in some ways as well. But thinking beyond, and that's because these places are so similar, we think about Earth-type life based on carbon, based on similar kind of metabolism. But thinking beyond the solar system, a lot of people have kind of done a bit of sort of hypothetical thinking about what other types of life could exist, and actually what falls within the remit of life. So people have talked about silicon-based life as a sort of alternative to carbon-based life, because it's got a similar number of bonds. But you would have to have really, really specific, really sort of specific conditions for anything like that to actually evolve. Whereas what we understand about the universe and sort of the planets that we're going to come into contact with is that we have a lot more similarity with Earth and with planets in the solar system than than these sort of like quite out there, very different forms of life that you might get. Within the solar system, I mean, there's been sort of hypothetical, very different kind of ideas about how life might work. So you might have seen the research that came out last year about the clouds of Venus and whether or not microbes could actually be living an entirely aerial life cycle in the upper clouds of Venus, where it's cool enough for life to actually survive. So I mean that kind of remains to be seen, but there's loads and loads of missions that have just been announced that are going to go to Venus and probe the atmosphere and look at different things about how Venus evolved and why it ended up so different from Earth, despite starting off so similar. So there's lots of different ideas about like really out there places and sort of setups that life could have. But by thinking about Mars and by thinking about the icy moons as well, so places like Europa and Enceladus, which is one of the tiny moons of Saturn, but both of them have these liquid oceans that are probably fed by hydrothermal activity, and they have these ice shelves above them. So we've got these kind of almost deep sea hydrothermal environments that are very similar to what I showed you very early on with the sort of deep sea hydrothermal vent systems. So there's a lot of environments just within the solar system that do really cross over with what we have on Earth, as well as before we start thinking about like really different setups that we could have.
I had a question talking about what you were kind of talking about what you were talking about there, but also picking up on what you were talking about earlier about nitrates and how they can almost replace the role of oxygen in a lot of different molecular processes. The question is about whether they just literally just replace oxygen or whether they create different complexes when they're bound together.
So nitrates in the context of the organisms that I'm kind of working with, they basically take the role of oxygen. So for all organisms, you have an electron. You have to basically pass electrons through a transport chain to create energy. For those of you doing A-level biology, that might sound quite familiar about the electron transport chain. But yeah, that happens in all organisms. It has to happen because electrons are the sort of source of energy for these organisms. So you need to get the electrons from somewhere. That's the electron donor. And at the end of that process, you need to give the electron to something else. So in our case, we're sort of taking electrons by oxidizing sort of organics in our food. And then at the end, the electrons being passed on to oxygen, and we're forming sort of CO2 as a sort of byproduct of our metabolism. In terms of these microbes, what we're doing is we're starting off with iron 2, the one that isn't rusty. And we're taking an electron from that's becoming iron 3, the rusty one. The electron gets passed through the microorganism, and then that electron eventually gets passed on to nitrate. So that's NO3, sort of nitrogen with 3 oxygen atoms attached to it. So one of the oxygen atoms gets knocked off that, and we end up with NO2, which is nitrate. And then these organisms, yeah, it sounds very complex, but basically these organisms, some of them can continue to do that and knock another oxygen off and another one. And eventually you end up with nitrous oxide, nitric oxide, and eventually it ends up as just nitrogen gas, N2, getting cycled back into the atmosphere. So yeah, it isn't quite the same as oxygen because you have these different steps that can happen afterwards, but it's performing the same role in that setup as a sort of electron acceptor at the end of the electron metabolism.
Great, thank you. I think it's quite interesting how whenever you're talking, it's chemistry, and then it's also geology, and it's also biology. It's all these different things that are coming together in all of your work. I wanted to move on a little bit and talk about that and your career and how interdisciplinary you are. I studied biomedical science as well as you did, and when I was looking at this iron cycle, I was like, oh, this feels like I've seen a similar thing in like the kidney, for example, and that you have all these cycles going on. Can you talk about a little bit how the things you did right at the beginning link into the things that you're doing now?
Absolutely, yeah. So you're absolutely right that it's quite strange how often you see sort of elemental cycles in the environment mirrored within complex organisms like us, or just within sort of cell biology. So yeah, that's a really cool thing, especially, yeah, like you say, the way that iron cycled in the body and stuff like that, and these different oxidation states of iron that you can get. It's also really tied into iron specifically. You'll find is right at the middle of a lot of the enzymes that we use in our own cell biology as well. So it's really got a fundamental link with sort of the way that cells function in our cells, but also in microbes as well. But yeah, the crossovers. So as I've mentioned before, it really helped me out in my PhD that I worked on the sort of ideas around knockout studies, where you basically take an organism, you have the organism as it is, which is called a wild type, and then you basically take one gene out of it, and that's the mutant. So by knocking this one gene out and comparing how these things grow, or whatever you're interested in between these two organisms, you can kind of find out, it's a way of finding out what that gene did. Because if you take it away, what goes wrong, basically, is is how you look at that. So that's something that's largely been developed through biomedical sciences, and kind of looking at the sort of human biology and physiology, and the sort of history of that. So that really crosses over. There's also just general techniques. So I mean, if you want to do the kind of microbiology type astrobiology that I do, then a lot of it is kind of looking at cells under a microscope, being able to operate those kind of things, being able to culture cells, which is something that you learn in sort of all range of biology degrees that you can do. So there's kind of, as long as you've got the kind of, I guess, hard skills in sort of lab skills and things like that, you don't have to be, this is the thing, you don't have to come out of your bachelor's with every single skill you need to then go for a career in astrobiology. But it really helps to have some of the kind of basics in how to kind of culture microorganisms, for example. You'd learn that on a lot of different biology courses. Even if you only have experience of sort of culturing sort of mammal cells on a petri dish, it's all the same kind of skill set that you build up through just having lab biological skills. And it's the same with my friends who work in the kind of geological or geochemical side of astrobiology. Most of them started off doing sort of pure just earth geology or geochemistry, and looking at how these different systems work. And then really all astrobiology is applying those sciences to a context off Earth. So yeah, I mean, whatever you choose to do, whatever your kind of passion is in terms of your, whether you really love biology or chemistry or anything like, or physics like that, there's lots of different routes into astrobiology. It's really just how you apply what you've learned once you get to the stage of sort of conducting your own research, really.
Yeah, you were talking about this astrobiology graduates in Europe group. What sort of people are in that group? Are there other biomedical science people in that group? Is it a really mixed bag of disciplines?
It's such a mixed bag of disciplines, and like I said, you meet all kinds of people. So at the moment, I'm serving alongside on the committee of this thing, which is entirely voluntary. It's something that we kind of do in our spare time, just because if you work in this kind of field, quite often you're so excited about it that you want to spend some of your spare time like getting people together to talk about it. So yeah, some of the people that I work alongside, some of them are microbiologists, but actually our current serving president started off doing a bachelor's in astronomy and just really loved space sciences, and then ended up working in a group where they had a bit of an astrobiology crossover with a microbiology department. She did a sort of crossover into a second degree doing microbiology, and now she does microbiology as a main thing, but she has that background in astronomy. We also have people who are sort of geochemists. We have people who are organic chemists and are interested in the origin of life. So that's almost a very different field, but it's kind of within astrobiology. Is you have people who are interested in how inanimate kind of chemistry, well, not inanimate, but non-biological chemistry, what is the dividing line between really complex chemical interactions and what we call life? And the answer is we don't really have a good definition of life. So there's a whole field of astrobiology around trying to find something that we haven't very well defined. But yeah, there's all sorts of people from all different backgrounds as well. In physics as well, astronomy and sort of cosmology. So astrobiology encompasses those as well, because if you're looking at hunting for exoplanets, everything you can find out about an exoplanet, we're now getting to the stage where we can start to probe the atmospheres of some planets that are orbiting other stars, which is incredible. It's something that I wouldn't have thought possible a few years ago when I started getting into this kind of stuff. But we're getting to the point now where we can start to detect atmospheres around sort of larger planets, and they'll get smaller and smaller and smaller what we can detect. But that's astrobiology, because if you can look at the atmosphere of a planet, it might have biosignature gases in it. And this all kind of falls within this interdisciplinary astrobiology atmosphere. So it's a great place to work.
Yeah, sounds like it. And it sounds like the rate of change in these disciplines is quite big as well.
Absolutely.
I want to throw some quick fire questions at you,
yeah.
which hopefully shouldn't be too hard to answer. And they're all about Mars.
Yep.
So on Earth, we have tectonic movements. Does Mars have the same?
So from very early on in Mars's history, it wouldn't have had the chance to have tectonic movement. It's a solid state planet, just because it's a lot smaller than Earth, which means that it cooled a lot quicker, just pure physics. It cooled a lot quicker, which means that it didn't have a sort of period in which these tectonic plates could have formed and then moved around in the same way that we still have on Earth. So Mars is a much colder place than Earth is today, because it cooled off too quickly for tectonics to take effect.
And I've got two questions. Does rain occur on Mars, or could it have occurred on Mars? Is there a different definition of what rain is? And can we grow plants on Mars?
Okay, so rain almost definitely did occur on Mars, like I said, in this really early period, the Noachian, 3 or 4 billion years ago. We would have had this warm, wet environment, and that would have had a water cycle that would include condensation of water in the atmosphere, raining down into the river channels that we can see existing today. But today on Mars, cold and dry, all you get is very high level clouds that are made of ice. So you never get rain as you would do to Mars, because the average temperature is about -20. So you never really get the chance for rain to occur today. And the second one was about plants growing on Mars. So we've already been able to grow plants on the International Space Station, which is a good start if we want to take plants to Mars to support our own human settlement and human habitation. But there's also a lot of active research where people try and grow, researchers try and grow different plants in Mars simulant soils, so soils that have the kind of analog similarity to what we have on Mars. And by all accounts, they've been quite successful. I know that there's actually a national Center for Potato Research in Peru that was doing Mars soil analog research to check whether these strains of really straight, hardy strains of potatoes could grow in Mars analog soils. So there is a lot of active research to do on that, but the likelihood is if we took soil to Mars, if we took plants to Mars, we'd probably use a bit of the Martian soil. We'd have to clean it up to take some of the harsher chemicals out of it, but we'd probably bring a lot of the nutrients that the plants need from Earth with ourselves.
Okay, well, I think we're going to wrap up there. So thank you so much. Just want to say thank you for your talk and for joining our series. I think it's really interesting to see your specific research and to hear about how you approach your work. So thank you.
Absolutely welcome.
Just want to say a little bit about next week. Next week we'll be joined by Lucinda again from the Royal Astronomical Society, and also our speaker will be Dr. Emma Ryan, who's going to talk about her speciality, which is engineering and space materials. So that's going to be a fantastic talk as well. I'm so happy with how all these talks are going. So thank you very much, Alex, and thank you very much, everyone. Sorry I didn't get through all the questions. Thanks.